I'll be talking about how we're integrating the ElysION into our current sample to answer workflows at the Nebraska Public Health Lab. So no conflicts here. So the mission statement at NPHL is dedicated to protecting the health and safety of Nebraskans through diagnostic laboratory science, technology, and education. So your first question probably is, what is a Nebraskan? So we're located in the center of the United States, and we're famous for corn, cows, and Warren Buffett. UNMC is located in Omaha, which is on the Missouri River next to Iowa. And so we're co-located. The public health lab is embedded at the medical center. And we're also happen to be right next to Nebraska Medicine, which is the largest hospital network in Nebraska. So access to a lot of samples. So for the public health labs in the US, a lot of sequencing came through the CDC PulseNet program. And so PulseNet started with pulse field gel electrophoresis and was used to track foodborne illnesses and outbreaks. In 2013, they started transitioning to whole genome sequencing on the MiSeq platform. And by 2019, all 50 states were started doing whole genome sequencing. For NPHL, in 2021, during the COVID-19 pandemic, they acquired a sample to answer platform called ClearDX by Clear Labs to automate sequencing of SARS-CoV-2. And then Clear Labs was able to upgrade this with Illumina so that we could also be able to sequence our bacteria pathogens for PulseNet. And so we've had the ClearDX system since 2021, and then ElysION arrived in December 2024. And so just a quick overview of what the two platforms give you. So ClearDX comes with a GridION, but just has two flow cell positions in there with an optional Illumina upgrade of what would be two iSeqs. It's on a Hamilton liquid handler system. It does require the internet. The kits are meant to be turnkey, so thus they're not flexible at all. They're meant just for the ElysION, but I have to say they do include everything, and when we say everything, well, everything except ethanol, even the inoculating loops to the bead bashing tubes to be able to do this, which does cut down on the setup time. We're able to bead bash, set the thing up in 30 minutes. And gives a runtime of around 27 hours for extraction, library prep, and sequencing. You get hit on the throughput. We're 8 to 12 samples per run between the two iSeq flow cells, and the cost is around $2,000 US per run. So for the ElysION, what we have in our lab is the MK1D setup. We currently right now have one device in there with I think in the coming quarter or two, we'll get two additional ones. Will give us a total of 3. There's also the optional P2 Solo, which I think they're displaying. Has the liquid handler as a TCAN. It can be connected or standalone. So currently, we're actually using standalone at the Med Center, more or less because I didn't put in the paperwork for the IT. But it's been working great with the only issues of updating and troubleshooting a little bit more work. It does use standard kits from ONT right now, so the same kits you'd order for RBK is going to work on ElysION, so you're really only limited by the available protocols. Setup time is a little bit longer with the bead bashing step and just putting everything together. We're having it at 2 hours. We could probably shorten this if we actually made things in the kits. And you can do up to 24 samples currently with the no-miss protocol with a cost of $1,100 to $1,300 per run. And that's assuming using a flow cell that costs $700. So the majority of the sequencing that happens at NPHL is probably 85% of that is through PulseNet. The other 15% is through another program through the CDC called RLN, so the Antimicrobial Resistance Laboratory Network. And so that's where we get some of our samples that are involved in the hospital-acquired infections. And so just looking at a subset of some of the bugs we sequenced, so Nebraska Medicine saw increase in KBC positive isolates in 2024. So we investigated it, and so the data on your right is data that we get through. So right now, all our data is going into a software package called BugSeq, and so we'll get serotype and the primary plasmid cluster. And so what we're looking right here is all the KBC2 positives over the course of from 2021 that all have the same cluster. And so what we have, so the first thing we did is look at the actual organism, so comparing data between, again, Illumina generated on the CLIA labs and Oxford Nanopore data generated on the ElysION. And this is the caveat here. When we first got the ElysION, the data was, it was hard-coded for high accuracy base calling. And so this data is all with the high accuracy algorithm, not the super high. But still, what you see is the tree topologies look exactly the same, and just highlighting some of the coverage drops. Like usually, what the CDC is asking for is 30x coverage for Illumina. Not as much guidelines for ONT. We're shooting for 50, 60x coverage for ONT. And then when we do the hybrid assembly, again, everything looks the same. And then they're clustering based off of, so the serotype 22 is in blue and the 112 is in black. When we then mash all this together, and I failed to mention it earlier, what we're doing, this is a pangenome analysis. So when we then take the Illumina, ONT, and hybrid captured data together and run a pangenome analysis, we are seeing all similar amounts of core genes and total genes. So everything is kind of matching up. And then we actually, if we take a closer look at that cluster, which is from the serotype 112, and no one can see this, but you'll just have to trust me. When we're looking at the SNP differences between Illumina, ONT, and the hybrid capture, see everything is pretty consistent within a distance of 1 to 2, and that's consistent through all the samples. We had one problem child that had 28 SNPs difference from the ONT, and looking back at it, has the little bit lower coverage on that sample. And again, so we're also then, can clearly see some clonality as well when we're comparing the SNPs. Again, another busy slide with then taking that same data generated from CLIA labs and the ElysION, looking at the plasmid. So first, the sizes of the contig that contains the KPC2 gene. We're getting more comparable size with ONT versus hybrid assembly. We're also looking at the replicon type using MobSuite and Plasmid Finder and getting consistent results between all three. And then we'll just take a highlight of these INC-P and INC-P6. And so again, working with Richard Stanton from the US CDC, able to use some of his software packages for aligning the plasmids. And so what we see is a little bit better alignment or fuller alignment with ONT compared to Illumina. And so just quickly to wrap everything up, what we're seeing is both automated platforms are generating coordinate data at the organism level. So I'm not really seeing any difference between Illumina and ONT. Again, the plasmid replicon types are agreeing. However, as probably obvious, is we're getting better resolution of the plasmid sequence using the ONT reads. And we're also looking at seeing both evidence of clonal transmission and horizontal gene transfer of the KPC2 encoding plasmids. And that at least within this context, so looking at these Citrobacter examples that I gave you, that our ONT-only bacteria assemblies do appear to be suitable for the hospital-acquired infection investigations. And with that, just like to thank everyone at the Nebraska Public Health Lab, UNMC, our partners at the Nebraska Department of Health and Human Services, and the US CDC. Thank you.